Plastic evaluation is our core expertise at Gradical. We help medical technology companies choose the right plastic for their application with the technical suitability, regulatory compliance, and improved sustainability. Learn more under www.gradical.ch/plastic-evaluation. Our guest today is Monir Azzouzzi. Monir is the founder and CEO of Easy Medical Device, a Swiss-based regulatory affairs consulting firm. Monir hosts his own podcast, the Easy Medical Device podcast, where I have had the honor of being a guest on material selection. Monir provides all kinds of regulatory affairs services, including the clinical evaluation, the topic of today's podcast. Here's our discussion. Hi, Monir, and welcome to the podcast.
Thank you for inviting me. Really appreciate it.
You're a regulatory affairs expert, and the topic of today's podcast is clinical evaluation. How does one do a clinical evaluation, and what is this?
So mainly a clinical evaluation is a document that is mainly created by some really expert on clinical evaluation to prove. So the objective of it is mainly to prove to the authorities that your product is safe and performant. So mainly safe means that it will not kill anybody, or that the risk of killing is lower against the benefits, or that the performance, which means that the claims that you are doing. So if you claim something about your device, that you have to prove it through your clinical evaluation, that it's really happening. So it's like when you have people say, oh, my product can do this or can do that. Okay, prove that to me. How can you prove that to me? Show me proofs of it. So mainly this is what a clinical evaluation objective is.
Mm-hmm. Okay, so can you walk us through how you would do that with, let's say, the example of a device? I'm doing let's say, an incremental device. So I have my next generation of a device that I already have on the market, and I do some improvements on usability and have some clinical benefits. Maybe I change also the material to a more sustainable material. How would you do the clinical evaluation in this case?
So mainly what you have to start with on the clinical evaluation is to really have a good understanding of the products and the scope of the product. So the description, as said, what are mainly the benefits of using your products? What are the different functionality, the different variations that they have, etc. So mainly we should focus on the product itself, not the previous generation for the moment. So we just focus really on this product. So we should have on this clinical evaluation all the description and understanding of it. Then we are planning within the clinical evaluation. We have two documents. We have the clinical evaluation plan and we have the clinical evaluation report. The clinical evaluation plan is a plan of how you will execute your clinical evaluation. So mainly you have to describe all the steps that you will be following, which for the literatures, for example, on which database you will be going to search for those literatures, what kind of keyword you will be using for that, how you will be considering a product as equivalent or not equivalent, etc. So you will have to plan. So here we just know the products and we plan now how I will do the search and do the exercise of the clinical evaluation. So it means that for the moment you haven't searched anything on the products. You are just planning for it. As soon as you plan for it, then you can start now to execute the plan, which means that you have to go to the databases that we talked about, and then to find out multiple information. The first information that you have to find out is about what we call state of the art. So mainly state of the art means, okay, we have your product that is doing certain things with some function, with some claims. What if we don't have your product? So what are the other alternatives that are existing on the market that are state of the art, that are like used by a lot of people? So it can be a product like a medical device itself. It can be also a drug. It can be also other alternatives. So mainly list me all the alternatives for that. And you have also to search literatures on those alternatives. So this is what we call SOTA, S-O-T-A, so for state of the art. And this search will be providing information about which solution exists on the literatures, what are the benefits of those solutions, etc., etc. So here you are just making a search of alternatives. When you have done that and you have really compiled all the information about alternatives, then now you are searching about your device. Many you say, what are the clinical evaluation, clinical information about my device. If you have yourself some data that are existing, then you can also use them. If you have, for example, executed a clinical investigation, then this is something that you can use to explain that I have tested my device on humans and here is the thing. If you have not done some clinical evaluation, you can maybe use some equivalent devices to say, here are the device that I will be using as an equivalent. And the objective of using an equivalent device is not to say, oh, my device is equivalent to this device that is already on the market, and so we are fine, my device is safe. No, the idea is to say, I will use this device as an equivalent because this device has a lot of literatures on the databases, which means that if this device has a lot of literatures, if my device is equivalent to this device, then the literatures that has this device can be used also for mine. So this is mainly the mindset. But maybe we'll look to talk a bit later about equivalence because it's also something that is really important. Then when you have done this research of existing data about your device, equivalent data plus state of the art, you have to kind of compile all this information. And it's why we say that maybe you should be like a medical writer because you have to read all the literatures. You have to segregate. I mean, what we call appraisal means you have to select if this literature really makes sense for your device or doesn't make sense, and you have to justify that. So you have, for let's say, 100 literatures that you have identified. Maybe 20 of them are not fit because it's not really talking about the type of device that you are having. Maybe 10 of them are not fit because they are in another field, not the field that you are using, etc. So then you are removing or filtering so at the end maybe you arrive with 10 literatures only that are remaining that really are on focus for your products. So what we are doing here is that you have all those literatures, you read them one by one, you then have to summarize them and pick all the information that have to be used to prove that your device is safe and performant. So this is mainly the objective at the end, to really prove that the device is safe and performant. So your literature data plus your own data plus the literature data of your equivalent device. If you have all this information, then you can make like a combo, if I can say, and then provide really a proof, evidence to the authorities and say, there is no possibility that the authorities are refusing my device because I provide so many data that, it's really impossible for them to say my device is not safe or is not performant. And then after you have done all this, you have to make a conclusion that your device, yes, is safe and performant. The risk is lower than the benefit. That you have also some tests done by the device, because you can also mention the bench test that you are doing about the device, not only the clinical one, but the preclinical one. So you have a lot of information to provide. And after that, if everything is finished, you conclude that your device is safe and performant, and you can then put it on the market. So here, I try to summarize it on step by step. There is a specific document that can really show that to you, which is the MedDev 2.74 revision 1, which is mainly the clinical evaluation guideline of how to do all the steps that we just talked about now. And for the ones that maybe are asking, those kind of this clinical evaluation can take maybe 2 to 3 months to compile. Because as I've said, if you have all the literatures to read, understand, and then you compile all this together, etc., it can take really some time to do everything. So if it's a simple device, maybe it's less time, but if it's really a complex device, it can take really a lot of time.
Okay. So, and you would do that basically on a paper basis. So you research the state of the art, you research literature for data for equivalent devices. So you're trying to fill data gaps that you do not want to test yourself from devices that are existing already. Maybe your device is so novel, or you're doing something that you have not found in the literature, that you have to perform a clinical test.
Exactly.
So how would you do that?
So if you have to perform a clinical investigation, first, you cannot do a clinical evaluation before you have executed your clinical investigation, because mainly the clinical investigation will feed the clinical evaluation. So you cannot just go in the other way. So maybe you can start a clinical evaluation. You will find out maybe that you don't have enough clinical data with your things. And then you have to go back. Okay, let's do now a clinical investigation to really gather data so that we can use for our clinical evaluation. So here you have really two separate things: clinical investigation on one side, clinical evaluation on the other side, are really two different things. and the clinical investigation feeds the clinical evaluation. So what you are doing mainly is that you have then to start to create. So you have to follow one standard. The standard is ISO 14155. Which is a standard that is explaining the different steps to follow for clinical investigation. A lot of companies don't want to go through clinical investigation because it's really a process that is long, costs money and everything. But what you have to do is that as soon as you want to do a clinical investigation, you have to decide first where you want to do that. Do you want to do that, for example, in France or in the USA or in Brazil or in Switzerland or wherever? And then if you want to do it in a certain country, you have to contact this country with also asking the authorization to do this clinical investigation. So you say mainly you are explaining to them why you want to do this clinical investigation, how many patients you are expecting, what are the outcomes that you are trying to get, etc. So you create really documentation to explain your intentions, because the objective of this the authorities are mainly that you have to prove that you will not torture people. So mainly you are not doing this just for your pleasure to do this clinical investigation for your pleasure. So you have mainly to explain why this is really something that is really important to be executed. If you do that in multiple countries, you have to do the same exercise with multiple countries also. So it's not like just one time that you are doing this exercise. Then you send this information, you wait for their approval to approve that mainly they accept you to do that. And if they accept, then you have to start now to recruit the patients that will be doing this clinical investigation. And just the recruitment of patient can take a lot of time also. Imagine now that you need for your clinical investigation like 200 patients. You will never have 200 patients day one of your clinical investigation. So you have maybe 5 patients on day one, maybe 2 days after you have 3 patients, and 2 days after you have 5 patients, and 2 days after, etc. So you see, to arrive to 200, maybe you'll need 1 year or 2 years. It is also what can be the tricky point. More you need patients, more it can take time just to gather those patients. Now you have find the patients, you have the right centers where you can do this clinical investigation. Then you are using your device on the patient. For example, if it's an implant, you have to go through a surgery, you make the implant inside the patient, and then we are waiting now to see if the patient is recovered and is working well. So the idea also of the clinical investigation, yes, is not that as soon as you do the surgery, you have done it, it's a success, it's over. You have also to do a follow-up. You say, okay, now day 0, we have done the surgery. Let's do a follow-up after 2 weeks. Let's do a follow-up after 1 month. Let's do a follow-up after 3 months. Let's do a follow-up after 6 months, etc., etc. And as soon as you have enough follow-ups and enough proof that the device was safe during the surgery, plus it's safe after the surgery, then you can gather those data to prove that, yes, we have done 200 patients, 198 patients had the surgery, 2 dropped off because they didn't want to do it at the end. Then 3 patients had an allergy due to the implant, so then they were dropped off, and we have now 190, etc., etc. So you are explaining mainly on your literature. You create a literature and you explain what happens during the surgery, or during the clinical investigation. And then you are concluding if your device is safe or not. And those data, clinical investigation data, are the most pure data, if I can say, because they are really done on patients and on your product. So it means that you are not trying to use some equivalent data, some literatures from other devices, some other things that are far from your products. No, here you are using really your product, which is really something that a lot of people can trust. It's not like you are testing some different products, some different patient, whatever. No, you test really your products with the patient that needs to have these products, and we have the result immediately. And this is mainly what people are really trying to get. Okay. The only problem, as I've said, with that, and it's a discussion we have always with manufacturers, we try to have as many patients as possible because it proves a lot that your device is fine, but they try to have as less patients as possible because it costs also some money and some time to then have the number of patients. So we try really to help to find the right number, but for that, you need what we call a biostatistician that will be really calculating how much is the right number of patients for such study.
So can you give an order of magnitude what a clinical test will cost on a device?
So we calculate that usually per patient mainly. So it depends first. Okay, it depends also where you are making that. If you make that in Europe or you make that in the US, it's maybe two different prices. In Europe, for example, what we try to get is like 15,000 euro per patient example. But as I said, it depends also of the device. If it's a device that doesn't need to be implanted, maybe it's less. If it's a device that needs to be implanted, maybe it's more. So it depends also of how you will use this device also. There are some clinical investigations that cost nearly nothing, because, for example, software that are using artificial intelligence, when you have to feed this software with images coming from databases, etc., to prove that there is a cancer or whatever, I mean, maybe it costs just the purchase of those databases or those images and nothing else. We don't need to see patients or whatever, because mainly those images are sufficient to prove that this is the image coming from a patient, etc. So you can have this kind of thing. Or you'll have then if you don't have enough images, then you have then to go through patients to search for those images also. So these are examples where there is no fixed number. It depends of the type of product, the invasiveness, the timing, etc. of the device.
Okay, so in the case of what we had before, 200 patients times 15,000 means 3 million for a clinical test.
Exactly, but this is times 15,000 for Europe. Maybe it's like 30 or 40,000 for US. So it depends also of that. So it can cost millions. Yeah.
Yeah. So that's also explaining why medical device manufacturers usually don't want to do it and try to argue equivalence with other devices. So
exactly,
we could go into how would I argue that this is basically the device that I had on the market before. So it's equivalent to what I'm doing. It has just some tweaks and some minor changes here and there. How would you argue that?
So mainly first, if your device is a class 3 device, so this is also important. If it's a class 3 device, mainly you'll have to go through a clinical investigation. There is no real choice, unless you have really good justification, which is this equivalence, this extension can be one exceptions, but mainly they will ask you to have really some clinical investigation. The other way is that if you have an extension from a device that is already on the market and already performing well, you'll have to show the differences between the both devices. Mainly, if it's a difference of material or a difference of functionality or a difference of whatever, you have to explain why this difference is not significant in comparison to the performance of the device, of how it is actually working with the previous version. Because why? Because mainly you are trying to use the clinical data of a previous device that is already on the market to justify that your device can also be on the market. The problem is that if your new device has new functionalities, the previous device didn't have that. So mainly what you can do is that they will maybe say you can use the previous device as your equivalent or for these functionalities, but for these new functionalities that are with the new device, you have to redo a clinical investigation, which means also that you have to maybe go through again search on patient, but this time you will not make the clinical investigation on the previous outcomes because those ones were already clarified. You will just make clinical investigation on the new outcomes and new functionalities of your device. And the point here is that you cannot really use your previous device as an equivalent if there are really a lot of differences between them.
Okay, so let's take the example. What I always get asked is, how can we make our devices or consumables more sustainable, for example?
Yeah.
And there are certain material alternatives that are bio-based, but basically chemically equivalent. So, for example, we have a bio-based material that has a much better environmental footprint, and I use this now instead of the material that I have been using before. So in that case, how would I argue that these two materials are equivalent?
So again, if we do equivalency, there are 3 criteria to look at. And there is a guidance for that, MDCG guidance for that is having a table and explain to you how you can do equivalency. The first criteria is technical aspect. Is the product technically the same, doing the same thing, used on the same patient, the same way, delivered the same way, etc., etc. Technical aspect is really important because then you can say, no, technically it's different, so we don't use that. But here, in your case, it technically is the same. The product is the same. Second is biologically. And this is where maybe this can be a problem for your equivalent device, because biologically says, is it using the same material? Does it have the same interaction with the same body? Does it have the same biocompatibility aspects, etc., etc.? So here, on the biological aspect, as it's a different material, there is a difference. Now, can you justify, because you can always do that, can you justify that this difference of material is not significant? Can you justify or have some proofs that even if the material is different, it's working the same, interacting the same, leachables are the same, extraction are the same, interaction are the same, etc. And then maybe, I cannot say for sure, but maybe you can go with that with notified body.
Yeah,
And
here
then the last
you would
part,
do the biological evaluation
exactly.
and prove that it's biologically safe according to let's say ISO 10993.
It's not only biologically safe. It's like, as I've said, that the interaction with the body is also fine, because mainly the idea is where on the body it will be used, and if there is an interaction here that can create some damages also. For example, allergies. Like, this material is fine, biocompatibility is fine, and the other is also fine. But there are some people that can be allergic to this kind of material without compromising the biocompatibility of it. So it can be having some problems. So mainly this is something that can happen. So you have really to provide proofs of all this. Yeah. So this is the second one, as I said, the biological. The third one is clinical. Mainly the product should be clinically used for the same purpose, the same disease, the same stage of problem, the same population, the same whatever. So these are the three: technical, biological, and clinical. If you can prove that all those three aspects are covered, there are similarities. As I've said, there can be some differences, but you have to justify that those differences are not significant. If you cannot justify that, then you cannot claim that this is equivalent.
Okay. Is that also, let's say, a risk class dependent? So let's say I change the material on my class 1 device, which I use in let's say in contact with the skin transitionary, like a few seconds or something like that, and I change it from polyethylene to polypropylene for whatever reason. I want it to be more stiff or more sustainable, or for cost reasons even. And then I do my biological testing, see that there's everything fine. I have evaluated it. Then I can argue that this is equivalent. And I just need to basically document that how I planned and then executed, that I can show the technical, biological, and clinical equivalency, and then I'm fine, basically.
Yes.
I still will need to do the literature research, the state of the art, and all that things.
So first, you have to do exactly the same for class 1 and for any other devices. So there is no problem about that. The only luck that you have, you are really lucky if you have a class 1, is the class 1 is considered as the lowest risk of medical devices. So it means that even if there is a contact with the body, it's not a contact with the body that is like a skin that is damaged or whatever, because otherwise it would be class 2A. It's not implanted inside the body. I mean, there is no implantation through a surgery, etc. So it's a really low risk class. So if you do this, what you have said, yes, it's fine. And more importantly, nobody will check, if I can say, because it's a class 1, which is called self-certified, which means that you have to do the exercise as written on the EU MDR, but nobody will check. So the only way they can check is if there is an issue on the market, then they can come and verify what you have done and if there is really an issue with your products. But otherwise, there is no notified body. The only people that can check, they are the competent authority of your country that can come and verify that your product is really following the EU MDR. But for a class 1, I never saw a company coming and, a competent authority coming and checking a company of class 1 devices. So you are kind of lucky if you have a class 1, because it's you have still to follow the requirements, but it's really the minimum requirements.
Okay, good. And so did I understand that correctly, that the clinical evaluation basically also encompasses all the biological testing? Credical is the plastic expert in medical technology and supports you in the areas of sustainable plastics and material compliance. Our course, Sustainable Plastics and Medical Technology, will bring your development team up to date with the latest developments in sustainable plastics. In addition, we offer the course Regulatory Aspects of Plastic Selection at the CUTS and the SCUT set, which provides guidance through the maze of regulations when selecting materials. More under www.credical.ch.
So no, you have to do. So we have two aspects. We have the preclinical study and the clinical study. The preclinical studies are the studies that you are doing in laboratories, animal study, for example, or bench tests that you are doing on the laboratories or whatever. These are information that, so the way to prove that clinically that your device is fine, there are 3 stages. The first stage is laboratory testing or bench testing. You test that through burning the plastic, for example, it's a plastic, or putting that in certain temperature, or verifying the material, the ductility or whatever. So this is first step. Second step, you can use this device on animals. So mainly you are going through an animal study where you are implanting the device on animal to see if there is any allergy, any problem, any misfunctionality about it. You have to select which animal. Is it a rabbit? Is it a dog? Is it a pork? It depends of the size of the device and the functionality. And then you have to decide that. Then if the details on the clinical, on the animal study are fine, then you can go then to the clinical investigation. This is mainly the phase. You don't go to the human before you can confirm that biologically and with the animal, everything is working well correctly also.
Okay.
So it's why you go with the animal study, and then you can go to the human study, and then this detail will also be provided through when you will ask for authorization of clinical investigation. You will say, I have already done some animal study. The results are completely fine. Here are the results. Everything is okay. So there is no danger normally for biological elements to receive this kind of products. So if you do all that, then you are fine, if I can say, for also the clinical evaluation, because inside the clinical evaluation, you have the preclinical part where you are writing what you have done, and then the clinical part where you are writing what you have done on humans.
Okay. So if you want to prove that your device is clinically safe, you first have to do like what you call bench test. I would call a reliability test.
Yeah.
So for example, like tension tests, bending tests, stuff like that. So stuff you can do in the lab.
Yeah.
Then you would certainly start, because also the biological evaluation says that you should start with in vitro tests or with the chemical evaluation. So basically, you start to dissolve your plastic, you look at the leachables, you have your extracts, and then put it on cells, do cytotoxicity tests or irritation tests on maybe artificial skin, and then you start to go to the animal testings, so irritation on guinea pigs or whatever. And then you move up basically the whole biological tests, and then you have these done with all the animal testing. That is the step where you can start writing your request for a clinical tests on humans. That's basically how this goes.
Exactly. Yeah, exactly. And as said, you can stop here. You can stop at the animal study or the biocompatibility study if you have enough clinical data with literatures, with whatever else. Because if you can prove that your product is safe also in terms of biocompatibility, you can say then no need to go to patients because it's a well-known device also. It's a well-known technology, it's a well-known plastic, it's an isoplastic, etc. So as soon as you are proving all that, then it's completely fine. Just one thing, because people are making a lot of mistakes. They think sometimes that the biocompatibility testing on animals should be done on the raw material. No, it should be done on the finished product, because this raw material went maybe through cleaning, through some operations or milling or whatever, and then it's changed the surface of the material. So here you have to test on the finished products and not to test on the raw material, because some people say, oh yes, this raw material is biocompatible. I say, okay, the raw material is biocompatible, but is your product biocompatible? Not the raw material, but your product. And your test has to be done on the product, the finished product.
Yeah. You know what my favorite example for that is? POM, polyoxymethylene, which is a widely used plastic. It's very has very good friction properties. Is properly stiff, used in all kinds of devices, and this is very biocompatible material unless you burn it during production. So injection molding temperatures are too high, or you sterilize it with aggressive conditions, and then formaldehyde is released because this is the monomer. You cannot get it out of the material, and then you have formaldehyde in your medical device, which is not biocompatible. So it really depends on your process and sterilization, and let's say all the history of your device. If you're actually biocompatible, it's a good indication that the material can be biocompatible, but you can still basically mess it up with your production process.
Exactly. It's important to know because maybe you have some suppliers or people that say, "Oh, don't worry, we have the proof and certificates that everything is fine and it's used medical grade, etc." But yeah, if there is some specific processes that can change the material, then everything is over.
Yeah. So I'm really interested to know. So I do this whole clinical evaluation thing, find my state of the art, and then I try to find clinical data for my device. First of all, if I do clinical investigation, so I do clinical testings on patients, am I obliged to publish this?
Normally, yes. I saw some that stopped the clinical investigation.
Mhm.
So as they haven't finished it, they haven't published anything.
Mhm.
But if normally you have done the clinical investigation, yes, you have to publish. And this is also the objective of having that within your clinical evaluation. Because if it's just done by you only, and you are compiling that, you can be biased because you say yes, you can say whatever to the data. I mean, you can say whatever about numbers. But if you are publishing that, then you should publish that in a peer-reviewed magazine where you have some other people that review and criticize and maybe accept to publish it because the content is fine. And this is high quality clinical data because mainly a peer-reviewed literature is a literature that mainly gets the approval not only about the writer but also about the other people that are reading that or approving this to be on the market. So even if you have negative results, normally you should publish that. But as I said, you have some tricks where you can stop your investigation before the end and prove that, oh, we had to stop because whatever, and then you don't publish anything, and it's fine. But mainly, yes, normally you have to publish positive or negative thing. But even during your clinical study, as I've said, if you have some negative data, but you have positive, you have 90% of positive, but some negative, you have still to inform about those negative information. You should not hide any data. And also, what people are looking, and sometimes we are looking at that also, who published that? Is it the company itself who has made this study, or was it done by a external independent laboratory? And then we can see also this should in a grade the quality of the publication. Something that is just like a blog publishing some data is 0 level. If something that is published by the owner of the device itself, maybe it's 1 level. And if it's something published by completely independent laboratory that has no connection at all and is not paid at all by this manufacturer, then it's a high grade of quality information. So you see here, you have also to look at that when you do a clinical investigation search.
Do you enjoy the podcast? Then subscribe and rate it on your platform. And join the discussion on LinkedIn. Your feedback and comments help us improve the podcast and bring relevant topics to you. Let's form a better world with plastics. Yeah, because what I was thinking is like the first one who is bringing a device to market takes on the responsibility and the also financial burden of creating clinical data for that device. And
exactly.
the fast followers basically have a massive advantage.
Unless it's a class 3. As I said, if it's a class 3, they don't have advantage anymore. Mm-hmm. Because if it's a class 3 device, you have to do a clinical investigation yourself. Mm-hmm. But if you want to use the clinical investigation of another company, like an equivalent device, because another company has done what you have said, you have to ask them authorization. So you have to sign a contract with them to say, yes, you authorize me to use your clinical data for my device. So this company will never give authorization to use their clinical data for their device. The only way it can happen like that is like, for example, you are part of a big group of companies like Johnson & Johnson or whoever, and they have multiple branches, and one branch wants to use the data from another branch, and then they have to make an agreement together. They are sister companies, so it's not like there is a problem. But this is a case where you can do that. But otherwise, for class 3 device only. For class 2A, 2B device, you can use the data without asking authorization. But for class 3 device, you have to ask the authorization.
Yeah, that means that that's just a thought that comes to mind, that IP protection, basically patent protection, is the only way to get your competitors out of your device. Because if you're creating all the clinical literature, spend millions on that, and then the fast followers just can do that without spending a dime, that would be pretty pretty
So yeah,
darn difficult for you.
so as I said, if it's really a new technology, yes, the IP can really protect you because they cannot use the same technology for their device, and then they cannot compare your device to their device. So it's impossible because mainly they
Mhm.
cannot do, or if they do, it means that they infringed your IP. But you have also a lot of devices that have no IP. If I can say, because it's common devices, it's already on the market since many years, and then this is more difficult for you to protect you protect that
Mhm.
because mainly you cannot ask anybody to say, oh, you should not use my literature or whatever.
Okay.
So this is how, yeah, you can try to protect yourself, but it depends on which case.
Yeah. What about cases where I would say like this is completely obvious that it's been used forever? Let's take the example of syringes. I don't know when syringes were invented, but it's certainly 100 years or something like that. How do you get clinical data on something like that is so obvious to like the
So
general public?
we have also on the clinical evaluation a concept that is called well-established technology, WET, W-E-T. So mainly well-established technology is a technology that is on the market since many, many years, and we have enough data to prove that everything is fine. There is a standard for it. Everything is there. So yeah, as long as you follow the recipe of the device, it's working. But you have certain listed devices that are on the EUMDR, for example, screws, plates, the braces, wedges, etc. A lot of things are listed, like if it is this technology which is also implantable or which is also used, etc. It's called well-established technology, and then this technology doesn't need to have too much clinical data because mainly we have enough data about that. You can also, and they are, as I said, they are listed, but there are also some definition of what is a well-established technology. So if you can prove that your device is a well-established technology that is used since many years, that have never any problem, then you can mention that. But if it's a well-established technology, you will have a lot of literatures also about that. So it's also something that can be used to say, you see, we find 500 literatures talking about that. You have all this and that. So you can also use that. And if you are a well-established technology, it's great because the notified bodies will not really investigate too much about your device, because as soon as you can prove you are well-established technology, yeah, it's obvious, as you've said, that the device is working well. It was manufactured by a lot of people, working correctly, etc., etc., and then you can be on the safe side.
I don't want to put you on the spot, but what are the criteria for well-established technology?
Mainly the idea is mainly that we have a long history about this device. It's not like a 20, 30 years. We have a lot of literatures about this device. The device has been proved to be efficient without any issue also on the market, without any big problem. So there are 3 or 4 bullet points of that. I have to look at my class because I'm doing training about that. But mainly this is that. So it's really the fact that it's used in with many things, and even there is no more IP, if I can say, because at the end everybody use that. So why are you having an IP for this?
Yes. Okay. Good. So thank you very much for the insights on clinical evaluation. I hope the listeners are a lot smarter now. I am certainly.
Sure.
So let's come to the more personal part. Tell us a bit about your career and how you got into regulatory affairs and what led you to found your own consultancy.
Yeah, so mainly, I was so I wanted to be a doctor at the beginning, a medical doctor, a surgeon. But in France, where I was studying, they had only 2 chance to be to pass an exam. After that, you have missed that. So, and you have what we call a numerus clausus, which means that you have just a certain number of people that can get through it. So 20 or 30 people out of 1,000, for example. So yeah, I didn't make it the first time. Then after that, I passed directly to biomedical engineering school, where I tried to learn more about biomedical, because I thought, okay, I cannot be a surgeon, but maybe I can help surgeons to treat patients. And then after that, the point was mainly I wanted to be in the R&D department, manufacturing a lot of products, etc., saving lives, blah, blah, blah. But when I have done my internship, on quality and regulatory affairs, which I didn't like at that time, but I had to pass an internship and it was the only one available. So I passed this internship and tada, I liked very much quality and regulatory affairs. And from there, yeah, I've done all my career only on quality and regulatory affairs for big companies, small companies, etc. And recently, so just before the COVID, I decided myself, and it was really a long thinking, maybe 1 year of thinking, decided myself to say, okay, let's maybe open my own company and do my own things. And it's when I started in 2019 to open my company, Easy Medical Device, where I'm providing all these consulting activities, authorized representative activities, trainings, and other also things that we are doing here. So mainly the idea was to try to help as much as possible all the small manufacturers, because we are not working with big ones. We are just working with really small ones to really be able to compete also with big ones and be able to be on the market and get certified. So this is mainly that. And to be honest, yeah, as I said, I didn't want it at the beginning, but as soon as you really fall on it, you understand that quality and regulatory affairs are connected to every department of the company. They are regulating everything. They are guiding everybody, etc. And I really like this way to teaching, to explaining, to try to stay compliant also. So it's why I really like to continue that way. And now, yeah, after 5 years now, I'm here and working still on that, and I'm really happy.
Yeah, so that's also my experience. These quality regulatory affairs positions, they usually start out with engineering or something like that, and then you get a foot into it, and quality regulatory affairs is like flypaper. You stick
Exactly.
and you stick and you stay there forever. It's like
Exactly.
my dad too. He studied electrical engineering and then went into purchasing, and then somebody needs to take care of that pesky quality, because I mean
Nobody wants.
nobody wants to, and quality is obviously purchasing, because quality comes from the supplier. And then he got stuck there. Like 95, he did his first ISO 9001, and he also tells me 2000 and 2001, 2002, ROS was introduced, and like this whole avalanche of regulations. So he was there when MDR was announced, and he said like, yeah, this is really an issue for small for
Exactly.
small and
For small
medium
company. Exactly.
companies. He told me basically, and he was like there on the dinner table and said, this is something. So this is not going to be good for small and medium companies. And yeah, here we are, and that's the top discussion. And for the last what is it now? 8 years?
8 years, yeah, around this.
8 years
2017,
now.
starting 2017, yeah.
Yeah. So to the last question, what advice do you want to give to our listeners about the clinical evaluation?
Yeah. So under the previous regulation, MDD, clinical evaluation was still needed. So it's not like something that we discover now with MDR, etc. No, it was still needed. But to be honest, there was less scrutiny by notified bodies about that. They checked it, but it was not like a big scrutiny. Now, the principal mistakes that manufacturers are doing is clinical evaluation. So the principal reason for failing a certification is clinical evaluation. It's not the classification or whatever, etc., or the audit of ISO 13485. No, it's not that. It's a clinical evaluation. So we had a lot of manufacturers that failed to get certification and were about to kind of stop their market in Europe and stay also in US. And we're able to help them to really understand that clinical evaluation should be done on that way. So do the clinical evaluation correctly and seriously, and 80% of your certification is nearly ready. Because mainly this is one of the big problem of companies. They think, oh, let's me take a template, let's purchase a template, let's fill the template, and it's fine. No, as I said, you have to be really understanding the clinical evaluation, understanding also medical writing. And as soon as you have done that, hallelujah, nobody can really contradict what you are providing there. But understand also that even if you have done a clinical evaluation, the notified body will come back, and I had that 100% for all the one that we have published. They will come back with non-conformities about your clinical evaluation, because they are not the expert of your product. So they try to understand, they don't understand, so they ask you to re-explain maybe this part, or to re-emphasize this thing, etc. So you'll get also some non-conformities from notified bodies about your clinical evaluation. If you don't get, I mean, you are really lucky. But if you get, no worry, it's normal. But clinical evaluation is really, you should spend most of your time during the certification on your clinical evaluation.
So maybe another question that just came to mind: when should you start the clinical evaluation in your development process?
So you have to start early to just have a plan, maybe not to execute, but to have a plan, to understand, for example, intended purpose. What is your product for? I have this experience with manufacturers where we are starting and they are starting to put 5 claims to say my products will be for that, for that, for that, for that, for that. And then when we start to make a plan for all this, we say, we need a clinical investigation. Then you have to find patients that have this disease and other that have this disease and other that have this disease. So 5 group of patients with 5 different diseases to claim all the disease that we are trying to cover. So here is mainly something that can be really a problem because at the end, you will be spending too much time about planning this, organizing this, etc. So my point is mainly start early to have these discussions because then it can reshape your view of the intended purpose. You can say, okay, we said 5, and at the end I had a customer like that. We said 5, and at the end I said, let's go for 1 only. So then we have less patience, less burden. I said, let's go for 1 only. When we have started to launch, let's go then for the second one, maybe later, etc., etc. So they wanted to make that on phases because they know that it will cost a lot of money to get, as you said, 2 million, for example, just for one thing. So if you have 5 different groups of people, it will be maybe 10 million for it. So then you need money. So instead of doing that, so then start with 1 and then increase slowly one after the other. But unless you have the money for that. But yeah, this is mainly the point that we have during our discussion to say, start early to make your company understand that, because I'm sure all along the way until you have to do the clinical investigation, they will have changed their mind 10 times.
Okay, so thank you very much for the insights. I summarize, put your priorities on clinical evaluation and start early. Is that your last words?
Exactly, yeah.
Okay.
I hope they will follow that.
Good. Any other few last words?
No, I said EUMDR is a new regulation. There are a lot of requirements. So yeah, the only last word that maybe it's important is if you don't understand EUMDR, try to get trained on it. And if you are trained and you still have some difficulty, use some consultants that are really working on that. I know it costs money. I mean, I'm a consultant, so I know it costs money. But to be honest, it will cost you less money than if you go yourself to a notified body with all the technical documentation and certification and things, and you'll fail, and then you have to repay again the notified body, etc. So it will be a big problem for you in terms of budget. So instead of that, pay a bit more maybe for a consultant to help you to verify that everything is fine, and then you'll be on the right side. But yeah, don't go alone if you are not experienced. If you have no experience at all, don't go alone because you will fail and it will cost you more money.
Mm-hmm. Okay, so thank you very much. This was Monir Azzouzzi from Easy Medical Device, and until the next time.
Thank you. Bye.
Still looking for more in regards to plastics and medical technology? Visit our website www.gradical.ch and inform yourself on the services of Gradical. You can also check out our market report on medical grade plastics on www.medicalgradeplastics.de. Or you attend a training regarding material compliance at CATS or at SCATZ. And of course, don't forget to rate and subscribe our podcast wherever you get your podcast. Let's form a better world with plastics.